In this video, we are going to explain the relation between immunity, mainly the cell-mediated immune response, and cancer. As you know, the immune system not only recognizes elements that come from outside the body, referred to as non-self, but also abnormal elements found inside the body, such as cancers, that are called modified self. As you can see here, a healthy cell is tolerated by our immune system. There is no mature T lymphocyte to kill healthy cells presenting MHC class 1 molecules associated to a self peptide. However, the infected cell with a virus or the cancer cell are recognized by cytotoxic T lymphocytes. These T lymphocytes destroy or kill the infected cells and the cancer cells. First, what is cancer? As a definition, cancer results from an accidental loss of control of cell regulation during mitosis, which leads to an uncontrolled proliferation or multiplication or uncontrolled mitosis. A tumor is the result of the uncontrolled multiplications of body cells in a certain tissue or organ following an alteration in their genetic program. Here we have normal cells. Some of these cells become altered. There is an alteration in their genetic makeup. Many mutations accumulate, mutations inside the genes, and results in the production of abnormal proteins. The abnormal proteins, are degraded into abnormal peptides and presented on the surface of the modified cells here. So we have cancer. Now, the rapid multiplication and the uncontrolled proliferation of the cancer cells forms a tumor. These cells can invade neighboring tissues, destroying the separation capsules or the separation membrane, thus causing metastasis. The cancer cells can migrate through blood and invade other body tissues. The risk for cancer increases with aging of cells, which can be accelerated by many risk factors. For example, genetic factors, behavioral factors, our lifestyle, environmental factors, family predisposition, and immunodeficiency. When our immune system is weak, we can have cancer, mainly if the cell-mediated immune response is absent or altered. The cancer cells do not passively wait for the immune system to activate. Certain cancer cells can develop a camouflage or even block the action of the immune defenses. Our defenses are faced with an invader which is difficult to target. The body cannot eliminate cancer cells because they proliferate very rapidly and overwhelm the immune system. And as you know, we have the classical treatment for cancer, which are the surgery, the radiotherapy, and the chemotherapy. But unfortunately, these procedures have serious side effects because they kill the normal cells and they kill the stem cells in the bone marrow, in the gonads, in the testes, in the ovaries, in the skin, the stem cells of the hair cells, etc. So these therapies or procedures have severe side effects. That's why we need a therapy that targets and that finds the right target, which is the immunotherapy. Immunotherapy is a technique, is a procedure, is an approach that finds the right target by stimulating and improving our immune system. One approach in the immunotherapy is that presented in this document. We can remove a sample of tumor from a patient. This sample contains cancer cells and specific cytotoxic T lymphocytes. In blue, the specific cytotoxic T lymphocytes, and in yellow, the tumor cells. Now, this sample can be cultured in the presence of interleukin IL-2, knowing that IL-2 is responsible for the multiplication and the activation of cytotoxic T lymphocytes. So, under the action of IL-2, cytotoxic T lymphocytes proliferate and kill the cancer cells in the culture. After 1 month, the culture contains only activated cytotoxic T lymphocytes. So we have activated cytotoxic T lymphocytes in the lab. The increase in the number of T lymphocytes can be injected, so these lymphocytes can be re-injected in the patient, so they attack the cancer cells and kill them. So this document presents the concept of immunotherapy, which is a hot medical topic. Other approaches used in immunotherapy to stimulate the immune system, we can inject, for example, the CGB vaccine against which is anti-tuberculosis. This vaccine is used to fight tuberculosis and to create memory for tuberculosis, but is also known for its stimulating action. This vaccine stimulates the cell-mediated immunity. So we can use it to fight cancer also. We can also inject cytokines like interleukin, which are capable of stimulating the immune system. But the disadvantage is that cytokines can cause high fevers and reactions similar to those caused by a generalized infection. Also, we can modify cancer cells in the lab in order to become harmless, and this by irradiation. Then we can re-inject the harmless cancer cells into the patient. This change should be enough to boost our immunity, but not too much, otherwise the immune cells may no longer recognize the original cancer cells. So we can say that the immunotherapy shows the potential advantage of the manipulation of the immune system to fight cancer. We can use the cell-mediated immunity, we can stimulate the cell-mediated immunity in order to boost our immune system and to fight cancer. Thank you.